Hello guys, so today we will be starting with the regulation of gene expression. Though I have sent you the link for this, but I want to do my own. So regulation of gene expression means you know that gene is something, okay, I have already told you gene is something that will express a certain thing. It will either express a certain character, okay, it will code for some character. And regulation means to regulate something. Even in our kitchen we have gas stove, okay, and we have regulator for that. It will either allow the gas to pass, or it will not allow but prevent the gas to pass. So similarly, regulation of gene expression means if gene is expressing certain thing, okay, if gene is coding for certain things, so to allow the gene to code that thing, or to prevent the gene to code or express that thing, okay, it comes under the topic regulation of gene expression. Okay, so regulation can be done in many labels. Okay, one, transcriptional label, when DNA is being transcribed to mRNA, okay, at that time also this regulation happen. Okay, sometimes it can happen during processing level, that is regulation of splicing. So during splicing of mRNA, that time also gene expression can be regulated. Okay, or sometimes it can happen when mRNA is transported from nucleus into the cytoplasm. At that time also regulation can happen. Or sometimes it can happen during translational label, that is when proteins are formed, these polypeptides are formed from mRNA. At that time also gene can be regulated, expression of gene can be regulated. So this is just the general idea. Okay, regulation of gene expression means either to allow that gene to express something, or to block that gene to express something. So that comes under regulation of gene expression. Okay, so there are so many example for that, but in our book or our syllabus, here it is given the Lac Operon. Okay, Lac Operon, it is one of the example, okay, where there is regulation of gene expression. Okay, similarly there can be so many operon, okay, but in our syllabus the Lac Operon is focused. So let's see now in Lac Operon. Okay, in Lac Operon, a polycystonic, lac here refer to lactose, okay, lactose operon. Lac means lactose. So in lactose operon, a polycystonic structural gene is regulated by a common promoter and regulatory gene. Okay, so here you can see here in the diagram. So in Lac Operon, okay, operon is something, first of all we need to understand what is operon. Operon is when there are polycystone, okay, polycystonic gene. What is cystone? I have already told you before in the previous video. So if there is polycystonic gene, okay, polycystonic gene, more than one cystone, but if there is multiple cystone, polycystone, but it is controlled by one single promoter and a single regulatory gene, such type of structural gene, it is called operon. Okay, operon is having more than one cystone, polycystonic gene, which is controlled by one single promoter and a regulatory gene. Okay, such type of thing, it is called operon. So here it is given Lac Operon. So a polycystonic structural gene is regulated by a common promoter and a regulatory gene. Such arrangement is very common in bacteria, okay, and it is referred to as operon. So there are so many operon. Here it is given tript operon, era operon, but we need to understand about the Lac Operon. Okay, now coming to the main point, that is the Lac Operon. The Lac Operon consists of one regulatory gene. So here you can see in the diagram, Lac Operon, suppose this is Lac Operon, okay, it consists of one promoter region and one regulatory gene. When I talk about transcription in the previous video, I have told you in some of the transcription unit, okay, there are some regulatory gene, extra gene just before the promoter region. Not in every transcriptional unit, but in some transcriptional unit there is one regulatory gene which is present just before the promoter region. So here in this operon also one promoter region is there, and just before promoter region there is one regulatory gene. Okay. So now this regulatory gene here, which is denoted by, okay, in Lac Operon regulatory gene is denoted by I. I here, you can see I here, it indicates inhibitor. Inhibitor means a substance that will inhibit something, substance that will prevent something. Okay. So this gene, it will prevent. This regulatory gene, it will not allow, but it will prevent something. Now what is that? What it will prevent, we will understand. Okay. Now here you can see the Lac Operon consists of one regulatory gene, the I, okay, which is denoted by I gene. Here the I does not refer to inducer. Inducer means to allow something, okay, but here I refer to inhibitor, it will block. And it also consists of 3 structural gene, because it is polycystonic, okay, 3 structural gene which is denoted by Z, Y and A. You can see here in the diagram, it is denoted by Z, Y and A. These are 3 structural gene. Okay. Now gene is something that will express something, okay, that will code for certain thing. So the Z gene code for beta galactosidase. This Z gene, this gene, it code for the enzyme beta galactosidase. Okay. Now because of this beta galactosidase, okay, this is responsible for hydrolysis or breakdown of disaccharides into monomer, that is galactose and glucose. It is because of the presence of beta galactosidase enzyme, okay, the disaccharides can be break down into monosaccharides. Okay, and this beta galactosidase enzyme, it is coded by this Z gene. Okay. Similarly, Y gene is their structural gene. It code for the enzyme permease, okay, permease, which increases permeability of the cell for beta galactosidase. Okay. In order to have this beta galactosidase in the cell needs to be permeable. So that permeable, it is facilitated by the presence of this enzyme permease, which is coded by this Y gene. Okay. Similarly, A gene, this also code for trans-acetylase. These are also enzyme. So these 3 structural gene that code for different types of enzyme. Okay. Hence all the 3 gene products in Lac Operon are required for metabolism of lactose, in order to break the lactose. Okay. In order to break that lactose or metabolism of lactose, all the 3 gene are responsible and all the 3 gene are necessary. Okay, because these 3 gene, it is coding for different types of enzyme that will help in the breakdown of lactose. Okay. Now, so these 3 gene are needed, okay, together in order to metabolize lactose. So this is the general idea of Lac Operon. Okay, what are there inside the Lac Operon? Okay, there is 3 structural gene that is coding for 3 different types of enzyme that helps in metabolism of lactose, and it is controlled by one single promoter region, and just before promoter region there is regulatory gene, and regulatory gene, it will block this operon to get transcribed. Okay, it will block this operon or this gene to get transcribed into mRNA because of this I gene. Okay. Now here Lactose is the substrate for the enzyme beta galactosidase and it regulates switching on and off of the operon, hence it is termed as inducer. So here though I is inhibitor, okay, I is inhibitor, it will block this gene to get transcribed into mRNA, but in presence of lactose, which is inducer, okay, in presence of lactose here this I gene, that is regulatory gene, okay, it cannot block further, it cannot block this operon to get transcribed further, only in presence of lactose. If there is no lactose, then this I act as inhibitor and it will inhibit this gene to get transcribed into mRNA, but if there is presence of inducer, that is lactose, okay, it will bind to this gene, okay, this site, and then it will allow the structural gene, this gene, to get transcribed into mRNA, okay. All right. So the repressor of operon is synthesized all the time from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. Now here you can see repressor molecule is there, okay. This I gene, it will code for this repressor molecule, this I gene, okay, because it is a gene, it will code or it will express this repressor, it will make this repressor molecule. This repressor molecule, it goes to the operator region, okay, from which transcription happens. It will bind to the operator region, okay, and when this repressor binds to the operator region, what happens is that RNA polymerase, the enzyme which is needed for transcription, RNA polymerase, it cannot polymerize this mRNA from this structural gene. RNA polymerase enzyme cannot polymerize these 3 gene into mRNA when operator region it is binded by the repressor molecule, and this repressor molecule, it is coded or expressed by this regulatory gene, I gene inhibitor, okay. So but in the presence of inducer, now here Lac Operon, in Lac Operon inducer is lactose, but if there is presence of lactose, okay, or allolactose, sometimes allolactose, okay, it also act as inducer, not only lactose but there is allolactose also. So if there is presence of lactose or allolactose here, what happens is that this inducer molecule, either lactose or allolactose, it will bind to the repressor molecule which is synthesized by this I gene, okay. It will bind to the repressor molecule, okay, this repressor molecule which is coded by this I gene, and when this lactose or allolactose, which are also called inducer, it will bind to this repressor molecule, then the repressor molecule get inactivated, okay, inactivated. Here you can see inactive repressor. So this will get inactive. When this will get inactive, it cannot bind to this operator region, okay. When this repressor molecule will be inactive in presence of lactose and allolactose, it cannot bind to the operator region. When it cannot bind to the operator region, it will not prevent RNA polymerase to undergo transcription, okay. So RNA polymerase can now bind to the promoter region and polymerize the mRNA from this structural gene, okay. But if there is no lactose or allolactose, if there is no inducer, then the repressor molecule which is coded by this I gene, it will directly go to the operator region and bind to the operator region. When it binds to the operator region, RNA polymerase will get blocked from okay, and that RNA polymerase that cannot polymerize mRNA from this structural gene. But in presence of lactose or allolactose, in presence of this inducer, when it binds to the repressor molecule, the molecule which is coded by I gene. It will bind to the repressor molecule, and when this bind to the repressor molecule, this molecule get inactivated, and if this molecule get inactivated, it cannot go to the operator region and bind here, so that now RNA polymerase can bind, that enzyme can bind to the promoter region and polymerize mRNA from these 3 structural gene. Okay, here you can see Lac Operon, Lac mRNA, it is getting transcribed in presence of inducer. But if there is absence of inducer, if there is no lactose or allolactose, this repressor bind to the operator region and prevent RNA polymerase from transcribing the operon. But in presence of inducer, what happens? This Lac mRNA, it is getting polymerized because repressor molecule cannot bind to the operator region. Okay. Here you can see Z is coding for beta galactosidase, Y code for permease, and A code for transacetylase. So this is the idea about this Lac Operon. Okay, here this gene it is acting as inhibitor. Okay, but this lactose or allolactose, it will act as a inducer. Okay, now here you can see regulation of Lac Operon by repressor is referred to as negative regulation. Very important. Okay, regulation of Lac Operon by repressor molecule which is coded by I gene, okay, in the Lac Operon. So regulation of Lac Operon by repressor is referred to as negative regulation. Why? You can get questions sometimes. Okay, so here we can see regulation of Lac Operon by the repressor is referred to as negative regulation, because when this repressor molecule, it is coded by this regulatory gene, okay, I gene, then repressor molecule, it binds to the operator region and it will block this gene to get transcribed into mRNA. It will not prevent, it is blocking, it is acting as an inhibitor, not inducer. That's why regulation of Lac Operon, okay, regulation of Lac Operon by the repressor molecule, it is called negative regulation, because it is blocking. It is not allowing, it is blocking. That's why it is called negative regulation. Okay, so yeah, this is all about Lac Operon. I hope you understood. Thank you, class.